Hello, everyone. My name is Dr. Annalise Gorensek-Benitez. You may call me either Dr. Gorensek-Benitez or some variation of my last name, or Annalise. Either is fine with me. And I'm your instructor for Chemistry BMB Biology 371L. Welcome. It's going to be a most excellent semester. Just a little bit about me. I am a passionate protein chemist. When I'm not teaching, I am researching in the Alexander Lab. My undergraduate work was at Furman University. I just completed my PhD in chemistry at the University of North Carolina at Chapel Hill. In my free time, I enjoy quilting and knitting and playing with my adorable guinea pig, Lillian. You'll probably be seeing pictures of her throughout the semester. Our teaching assistant for this semester, she hasn't made an appearance in this video. You can see her right here on the syllabus. Her name is Lindsay Leisure, and she will be assisting us throughout the semester. So let's get down to business. Our lab is located in Wake Downtown 1905. So it's the lab here at the end of the hallway on the first floor, lit up very nicely in that photo. This is a 1.5 credit hour lab, in case you didn't know. So we meet for 3 hours and 45 minutes on Wednesdays from 2:15 to 6 p.m. Now, most weeks we will be done before or by 6 p.m. However, especially during labs 4 and 5 and 3 as well, we may go over time because as student scientists, you're responsible for your work, you're responsible for your samples, and so if necessary, we'll need to stay to get the job done. And so just to be on the safe side, I would not recommend scheduling any meetings between 6 and 7 on Wednesday evenings. Just a helpful hint. So for the attendance policy, so one thing that makes this class unique is that the whole lab is a single process, or the whole semester. So each lab builds upon the previous lab. And so if you miss a week, that's kind of a big deal in that you're missing a big chunk of the lab, and it's something that the next week depends upon. But it also makes it pretty hard on your lab partner. So there are only a few instances in which an absence from lab can be excused. So the first is illness, in which case you need a doctor's note. So last semester I had someone have strep and someone have come down with influenza B, which is why I have a protein model of an influenza B protein in green and brown, fighting a human virus fighting protein in pink. I thought that was kind of neat. So you need a doctor's note. Sometimes family emergency comes up. You may also have an official university activity that you need to participate in. For either of those instances, that will require a dean's note. I know that many of you guys are seniors, and so some of you may be going to conferences with your research, or maybe you have a grad school or a professional school visit. In that case, I would need a note from your academic or your research advisor. And starting with the third week of lab, we'll have a lab report due every single week. So in this case, if you have an excused absence from lab, your report will be due the following Sunday at noon. So just so you know. For those of you who have learning disabilities and need accommodations, specifically for the midterm and final exam, please contact the Learning Assistance Center within the first 2 weeks of the semester, and we'll make sure you're taken care of. So let's talk a little bit about lab safety. All right, so you guys are WAKE students, you've had several labs before, and you've been given the lab safety talk. So I'd like to approach lab safety in a creative way this class. So when I was in graduate school, this meme came out with this Japanese cat named Pusheen. And on the left, this is the original one. And you can see Pusheen is in this adorable pink lab with a pink periodic table. But clearly Pusheen is not implementing proper lab safety protocols. And somebody on Reddit called Pusheen out on this and was like, hey, he's not wearing proper PPI, there's no lab coat, chemicals are spilled everywhere and they're not labeled. Pusheen is clearly pushing the limits of lab safety. And so as a result, someone modified the Pusheen meme and then posted it on the internet. And here you can see that Pusheen is following acceptable limits of lab safety that would qualify her to work in 371L. So you can see she is wearing a lab coat, lab goggles, and lab gloves, at least lab gloves when you're handling samples. So all of those are required for 371L. The one exception, because apparently Pusheen doesn't have legs, that Pusheen is not wearing long pants. So if you're wearing leggings, these need to be leggings that cover your mid-calf, as well as closed-toed shoes. Although Pusheen is not wearing those items, you are expected to wear them for lab. No shorts, no mid-calf leggings, no sandals. If I see you coming to lab in unapproved clothing, I'm gonna have to send you back to your room to get proper clothes. And I know that taking the shuttle back to campus is a pain in the butt, and I don't want to make you do that. So please, please, please wear acceptable clothes to class. If you're concerned, bring some extra clothes, stow them under your bench, and now you have them in an emergency. I know I've kept lab shoes in lab before, so just an FYI. So you're going to be wearing gloves throughout the semester. However, you're going to be keeping track of your notebook on an eLab notebook. More on that later. So you'll want to remove your gloves whenever you're touching your computer because you don't want to contaminate it with lab samples. And then you'll also notice with Pusheen that from the left, she's not cleaning up her mess, but on the right, she is. So this also goes for our lab. You're going to be making messes some weeks, specifically weeks 4 and 5. And we're going to be purifying protein, and you're going to be keeping track of 5 different things at once. So it's really important to clean up as you go, but also clean up before the end of lab because the S in teaching assistant does not stand for servant. And both Lindsay and I would require that you clean up your mess before you leave lab. And then finally, I've hammered it into your head a few times by now, but in this lab, this is each week experiments builds upon the previous week. So your samples that you generate in lab 3, you're going to need for the rest of the semester. So talk to either myself or to Lindsay for instructions on where to dispose each container of liquid and solid waste. And it's also going to change from week to week. So that way we'll make sure you're not throwing away your sample. And if you laugh and say, oh, Dr. Gorensek-Benitez, we can figure that out. I had people throw out samples 3 times in my class last term. So check before you dump. That's the best advice I can give you. So talking more about the practical setup of this class, we are going to be using Google Classroom as the online interface. So before classes start, you'll be emailed an invitation to access the Google Classroom, but I also have the settings, so if something goes wrong with the email, you can access the course by going to www.classroom.google.com and sign in through your Wake Forest Gmail, and you can use this passcode to get into the class. I highly suggest if you have not access Google Classroom yet, that you do it immediately after watching this video. And that's because all classroom materials are going to be posted on Google Classroom, where you also submit all your grades and receive all your graded feedback through this website. As far as my email policy, I have a rule that I will respond to all student emails within a maximum of 24 hours of the time they are sent, at least emails that are sent between the hours of 9 a.m. to 5 p.m. Monday to Friday. For what happens during business hours, it's pretty self-explanatory, but if you email me at 3 p.m. on a Saturday, you might catch me at a time where I'm staring at my phone, and if it's an easy question, I might email back instantly. However, under this policy, the latest at which I'll get back to you is Tuesday at 9 a.m. because that's 24 hours from 9 a.m. on Monday, which is the closest business day to Saturday. So I'm teaching this lab because one day I want to pursue a teaching-focused career in biochemistry. I enjoy talking to you guys about the material. I like helping you, answering your questions. If I didn't want to do that, I wouldn't be in a teaching postdoc. So I highly encourage you to come to me with any questions you may have, whether that's shooting me an email and asking me questions that way. But I do have a weekly office hour in my office at Wake Downtown from 10 to 11 a.m. on Mondays. My office number is Wake Downtown 1613. So I'm in the joint office with myself and the other postdoc, Dr. Surya Banks, and we are right next to the Interfaith Meditation Chapel, which is on your way to lab. And finally, if you have a question that's too complicated to be answered by email and you can't make it to my office hour, just shoot me an email and we can set up a time to meet. I'm more than happy to meet with you outside of class. Some miscellaneous things. Be sure to bring your computer to class every week because, as we'll talk about next, you're going to be keeping track of your data in an e-notebook. So be sure to read over the appropriate lab materials to come prepared to lab each week. When in doubt, ask. And then finally, never throw anything away without asking first. I know, again, I keep saying that, but trust me, you don't want to throw out the wrong part of your sample because then you won't have anything forward going for the next week, and that's never fun. So we'll talk a little bit more about the eLab notebook now. So in past classes, you may have done a traditional hardbound lab notebook. Maybe, I know my sister who went to NC State had a formal online notebook form on a website on the internet. The way that we're going to keep your lab notebook is through Google Classroom. And I'll show you how to operate the notebook through Google Classroom in a second. But in terms of a lab notebook, the content is the same as what you would put in a hardbound notebook, with the goal being that if somebody wants to purify bovine heart lactate dehydrogenase, they should be able to take your notebook, repeat your experiments, and get similar results. So for each day of experiments, you're going to have to include the protocol, so for what you did each day, and then fill the protocol in with any data that you generated. So your Excel graphs of your activity assays, your purification table, how many figures you may have generated, they need to be there. But you also need to include things that you changed about the protocol, observations you may have made. That was the wrong color, or it made a bubble and it shouldn't have. That's kind of strange. You also need to keep track of things like your dilution factors. And then finally, for every type of calculation that you make, you need to include at least one example. So you don't have to write out every single calculation that you made in your lab notebook, but if you have to calculate the enzyme activity of 16 different samples, you'll want to include your final answer in your lab notebook, but you don't have to write out the equation for every single set of experiments. That's a little over the top. And then I have some more information on the specifics of a lab notebook, but I'll let you read through on your own time. But for now, let's take a minute. Let's hop to the Google. All right, so here's our Google Classroom interface. So you can see it's the Advanced Biochemistry Laboratory for Spring 2018, the Wednesday afternoon section. And so if we start off in the stream, so when you log into the classroom, this is what you'll see. See, I posted some videos as well as a practice problem set for pH. That'll be helpful for you to do those practice problems to help you out with pH. But this is where you'll see assignments as well as turn them in, and where I'll create drop boxes for you to turn your lab reports and your pre-lab assignments in. So what you'll notice is in the About tab, we have a class drive folder. And so in the class drive folder, this is where you're going to set up and maintain your lab notebook. So you can see here that I've created 2 theoretical lab folders. And so if I click into my lab folder, so Annalise's lab folder, this is where I'll create Microsoft Word or Google Word, the equivalent documents for my lab notebook. So it's up to me whether I want to have one running document and put all the labs in there, or if I want to create individual Word documents for each lab. The same goes for Excel spreadsheets that contain my data. But what's really important is that before you leave lab on that first week, that you take your folder that has your lab notebook in it and back to my drive. Okay. Sorry, guys. Messed up a bit here. We go into the classroom folder. Okay. That's last semester. Okay. But the point is that if we go to the folder, you right click and you see share. Okay. You need to make sure that the people that are on your settings are yourself, your lab partner, me, the instructor, and then Lindsay, your TA. All right, I'm not sure exactly how the permissions work. If students add things, you need to make sure that for every document you create, Lindsay and myself have access. So that way, when Lindsay's going through and grading your lab reports, she can go check your lab notebook and make sure everything is kosher, because your lab notebook's going to count for like 5% of your lab report grade. So now you know how to do that. So let's get back to the syllabus. So now that we've covered lab notebooks, you're also going to be writing a total of 10 lab reports throughout the semester, with the idea being that your lab reports will come together to make your final paper. So throughout the semester, Lindsay and myself will be giving you feedback on each of your lab reports. And the goal is that you will take that feedback and immediately apply it to your lab reports. So at the end of the semester, when you'll see, when I show you the grade breakdown, which 35% of your grade is due in those last 2 or 3 weeks, that way you'll have already done a lot of editing, so you'll have an easier time writing your final paper. Now, I'm not going to go into detail now about the lab report and the final paper. If you want, you can take a look at it now, but we'll talk more about lab reports next week and then final papers later in the semester. So let's talk a little bit more about the way that this class is set up. So we only have 3 hours and 45 minutes. And for some of these weeks, we're going to use the whole 3 hours and 45 minutes because it turns out columns take a long time to run. You'll learn that very quickly. And so one of the approach that I decided to take this term to facilitate a better use of our lab time is that we'll complete the pre-lab lecture ahead of time. So the way that's going to work is that each week I will post a video like this one and like the buffer video that I posted, have posted on the Google Classroom, and it will essentially contain me giving a lecture about about the material for next week's lab. And so I'll be talking and then I'll be filling in a few pages of skeleton notes. And the idea is that I'll provide the blank for the skeleton notes and you can fill in the notes along with me. And you can take the opportunity to pause when I tell you to pause and practice the practice problems that I give, but you can also rewind and listen again to things that I say if you didn't catch it the first time. So you're that way, instead of sitting and listening to me talk for an hour in lab before lab, so instead of having 3 hours and 45 minutes, we have 2 hours and 45 minutes and you're confused about the calculations and you just want to get out. That way, if you're a nocturnal person and you want to watch the pre-lab video at 3 a.m. on, Monday or whatever, so be it. You can complete it on your time. You can watch the lecture. You can watch the lecture at your pace, and then you can fill out the notes as needed. The other part of this is so it's really important that you watch the pre-lab lecture and you read over the lab handout before lab. And one of the ways that you can help assure me, or you can assure me that you have done your pre-lab prep, is that I do a pre-lab assignment. Now, this is a short assignment. So this is meant to take approximately 15 to 30 minutes, no more than that. And it contains anywhere from 2 to 4 questions, depending on the week, where I'll have you answer a combination of conceptual and also conceptual problems and also calculations for the week. And there will also be a slot where you can ask questions. And so then what I'll do is that's due 4 p.m. on Tuesday before lab. So then I'll turn around, I'll read over your answers, and then I'll take any questions that you asked on that sheet, as well as take note of problems that people are tending to miss. And then the next day we meet for lab, I'll start off by going over any misconceptions or any questions that you all have, and then we'll take the time. Then we'll take a few minutes as groups for you guys to work through the calculations that you're going to be using in lab. So I'll give you some extra time to have that practice. So when you've made your measurements and you're analyzing your data, you won't get stuck. Because some of these calculations, specifically in like lab 8, they get a little tough. And so this will make sure that you have an easier time during lab, and it'll also ensure that we're not wasting time by listening to me gab. All right. So the way that the grading will work throughout this term is that the pre-lab assignments, they will count as a completion grade or as a completion grade. So I'm not going to grade them for accuracy so long as I see that you guys are putting an effort and completing them on time. So because this is more, I want the emphasis to be on learning, making mistakes, and having the opportunity to answer questions. And I won't change that into being a grade for correction unless I notice a significant and sustained decrease in the quality of the answers I'm getting. So that will count. So those pre-lab assignments, as long as 2 homework sets, which are denoted in the schedule, will count for 8% of your grade. So a significant portion of your grade is actually going to come from a completion grade. So make sure you're completing those assignments and make sure you're putting 15 to 30 minutes in. Now, one thing to note is that 45%, so almost half of your grade, is coming from your lab reports. And so those are 10 reports that are going to happen over the course of the semester. And so with those lab reports, again, you're going to be getting feedback. So the idea is those lab reports will be taken, will be synthesized, and then added, and then with a introduction added, will constitute your, oops, a little crazy there with the with the arrows, but will constitute your final paper. And again, so the final paper, so if we look at the schedule, your final exam is the second to last week, your final paper is due the last week, and then the third to last week, you have lab report 11, and then a draft of the results and materials and methods section of your last lab report. So those last 3 weeks contain almost 50% of your grade. So it's of utmost importance that you are taking those lab reports, you're looking at the feedback that Lindsay and myself give you, and you're editing what you wrote. So when it comes time, when you're writing up your final paper and you're studying for your final cumulative exam, yes, it's cumulative, remember that, so that you can focus more on those last 2 aspects. And the writing of the final paper is going to be a lot easier if you work on it more in the beginning. Again, you also have a midterm, which is 10%. Both the midterm and the final are going to be in-class exams. And then finally, your lab performance and citizenship accounts for 2% of your grade. So that's not performance in terms of, oh, I have the purest protein, I get a 2%, I get a full 2% of that 2%. No, I mean, are you working well with your lab partner? Are you being considerate of your lab mates? And are you cleaning up after yourself? All those things will count towards and will give you a good lab performance and citizenship grade. All right, so now that we've covered the logistics for the lab, I want to talk a little bit more about the content of this lab and why this lab matters, and a little bit more about why it's a credit hour and a half. So what's the point? What do the drugs methotrexate, penicillin, and sildenafil, also known as Viagra, have in common? All these drugs are small molecules that target enzymes in the human body. So methotrexate inhibits the enzyme dihydrofolate reductase. Dihydrofolate reductase is one of the enzymes that's in charge of making precursors for your DNA bases. So if you use methotrexate to shut down DHFR, cells aren't going to grow and divide anymore. So methotrexate kills rapidly dividing human cells. Therefore, it's a common chemotherapeutic. So it's one of the reasons why people lose their hair, why chemo patients lose their hair, because you're targeting the rapidly growing cells in your hair. Penicillin, anybody who's undergone chemo, or somebody like me who takes methotrexate in low doses to help with autoimmune inflammation, methotrexate hits home for a lot of people. If you ever had a cold and you've had to take an antibiotic to combat like a sinus infection, penicillin's relevant to you because penicillin inhibits bacterial enzymes that build cell walls. And then finally, sildenafil, I actually included this one because it's an enzyme that inhibits it's a small molecule that inhibits the enzyme PDE5. And so sildenafil was actually put on the market. It was tested as a blood pressure drug and was found out to have a side effect that you've seen advertised on TV that turns out to be a lot more lucrative than a blood pressure drug. So all of these small molecules are inhibit enzymes. But before these molecules could be tested in organisms and eventually humans and then put on the market, some lucky biochemist had to go and purify the enzymes, so DHFR, PDE5, whatever, isolate them, and then characterize the interaction between the drug and the enzyme, and then look at the kinetics and the mechanism of how that inhibition worked. And before you can do that, you have to figure out how on God's green earth you're going to purify the enzyme. So that oftentimes requires going into the literature, digging out a paper, and reading between the lines to figure out what another scientist did. And oftentimes, other labs might include instruments that you don't have in your lab. So you have to switch it up and modify it. And so in this lab, we're going to be doing something very similar, and that we're going to be looking at the enzyme lactate dehydrogenase. It's involved in glycolysis. And so for that reason, there have been several drugs that have gone through clinical trials that were developed to target cells through inhibiting glycolysis by inhibiting lactate dehydrogenase. Now, it turns out that it's not really easy to find human samples to isolate LDH from. That's pretty unethical. But it turns out that cow heart or bovine heart is pretty easy to come by from our local slaughterhouse over in Yadkinville. And bovine LDH has 98.5% sequence similarity to human LDH. And so we'll be working with bovine LDH as a model for human LDH. And so over the course of the semester, we're going to be mimicking the work of biochemists by taking a protocol for purifying and characterizing LDH and seeing how well it works, what we would change, what we would do differently, what works, what doesn't work, and then commenting on it in the scientific journal article at the end of the semester. So what is this going to look like in terms of our day-to-day life in 371L? So as student scientists in labs 1 and 2, we're going to brush up on basic biochemistry lab techniques. So how do we make a buffer? How do we make a dilution? How do we monitor LDH activity and see how well it works? We need to make sure we can do these things and do them well so we can make accurate measurements throughout the rest of the term. During lab 3, your TAs, thankfully, will grind up bovine heart. Say thank you to your TAs for doing this. And then we will extract LDH from the cow heart mixture. During labs 4 and 5, we're going to take that cow heart mixture and we're going to purify LDH from all of the other cow heart proteins using 2 different types of column chromatography. And then for labs 6 through 10, we're going to elucidate the different characteristics of LDH and confirm that it's in fact LDH. So first, we'll look at the Michaelis-Menten kinetics of LDH and then see how it responds, and we add an inhibitor. Then we'll, in lab 7, we'll look at the size and the oligomeric state of LDH. So thinking about the quaternary structure, is it a monomer, a dimer, a tetramer, or something of even higher structure? Through labs 9 and 10, we'll be confirming the size and the identity of LDH. And then in labs 8 through 10, that's when we'll sit down, we'll look at all the steps together and all of our evidence and think deeply about the protocol and figure out which steps work best. Would we change anything if we would do it again? In lab 11, we're going to take a bit of a breather. And this will be a bioinformatics lab where we'll look at the structure of LDH and take a deeper look at how it facilitates the interconversion between lactate and pyruvate. For our lab, we're focusing, however, we'll be measuring the lactate to pyruvate reaction. And then finally, in the last lab, unlike lab 6, we were going to be monitoring LDH inhibition in vitro. In Lab 12, we're going to be watching LDH inhibition in cells. So you get two different looks of how LDH, or two different ways that biochemists look at proteins in dilute solutions and then also in cells. So you're getting two different aspects of biochemistry, which is really kind of neat and something that we do only in this biochemistry lab. And my goal is that throughout the course of the semester, you'll have a better appreciation for how small molecule drugs that inhibit enzymes, as well as protein-based drugs called biologics, are discovered and isolated, that you'll get to add different biochemistry lab techniques to your quiver if you're someone who is interested in research. And also, if you'll learn different lab techniques and critical thinking skills that may help you in research somewhere else, or an internship, or even in your job after graduation. And so, again, remember, this is not your average biochemistry lab, and there's at times where you may feel like you're just following a protocol. But remember, this protocol we're giving you, we're doing it because it's feasible to do in an undergraduate chemistry laboratory. It's not perfect. And so throughout the term, it's of utmost importance that you have your thinking caps on and you're constantly thinking about how the protocol could be improved and what you would do differently if given the chance. And so you can take a minute. I have the overall learning goals for the course down here, right over here. Take a minute to read them. I don't feel the need to read over the list because we've included them indirectly in this discussion of the course. And then finally, before we go, I want to do a breakup, or I want to show you a breakup of the lab and how the effort for this lab will be distributed. And so if we look at this drawing here, my apologies for it being a little crude. If we graph effort as a function of time, what you'll see is that labs 1 and 2 are pretty low effort and won't take a lot of time. But once we hit lab 3, that bovine heart extraction lab, through lab 6, where we're running the Michaelis-Menten kinetics, those labs, they're going to be challenging, especially labs 4 and 5, where we're running columns. They're going to take a lot of time. Those are the labs that are going to be the most likely to go past 6 o'clock, if we do have any labs that go past 6 o'clock. And so just be ready for those weeks. Once we get past lab 6 and we take the midterm, and then we hit labs 7 through 12, things are going to get easier. There's going to be less to do in lab, and although there may be some weeks that go long, there'll be more downtime. But one thing you do need to take note is that once we get to labs 10, 11, and 12, that's when 35% of your final, or the last weeks, and then into lab 12, labs 11 and 12, that's when like 35% of your grade starts to be due. So make sure that when you get to the end of the term, you've been keeping up with your work, so that part of the semester will be less of a load. And with that, that just about covers it. If you have any questions, please feel free to shoot me an email, and I look forward to seeing you in class. Have a great day.